Well, thank you very much, President, for the huge honor of giving this prize lecture to you this morning. I'm going to talk about small RNA, antiviral defense, epigenetics eventually, and the link that brings all of these topics together. I'm going to divide my talk into a number of sections this morning. So first of all, I'll give you a little background to how we came to work on RNA silencing. I'll then talk you through the various implications of the mechanism for our understanding of antiviral defense. And then towards the end of my talk, I'll segue into the topic which is our major preoccupation at present, which is epigenetics. Now, I'd like to tell you that you are awarding the prize medal this year to someone with enormous foresight and great vision. But I have to be honest with you. In terms of our work on RNA silencing, that is not the case. So this is a sorry story of blatant opportunism and massive amounts of luck. So we didn't start out, as it were, to investigate RNA silencing or indeed to do work on virology at all. I was not trained as a virologist. I was working at the Plant Breeding Institute, and the breeders there were screening their germplasm for susceptibility to virus infection, looking at tens of thousands of plants, and they wanted a test that was more reliable than the ELISA tests that they were using at the time. So they asked me whether or not I would develop a diagnostic test for plant viruses. And I was able to do that for them. And that then stimulated me to start thinking about more imaginative things that one could do with the cDNA clones that we had generated, or cDNA clones in general about plant viral sequences. And so then I started working on various virological phenomena. There was one in particular that interested me, which was how non-coding RNA components of plant viruses could stimulate the formation of rather specific symptoms on the infected plant. This is one example of a story that we followed up. We tied it down to a few nucleotides within this non-coding RNA, which accounted for the fact that this virus could induce a rather beautiful yellow chlorosis on infected tobacco plants and a lethal necrosis, which is less attractive, on infected tomato plants. So a highly specific symptom on different plants we could tie down to particular nucleotides in non-coding RNA. Unfortunately, we hit in the sort of mid-80s a bit of a roadblock there. We didn't know how to take that project a little further. I'll return to this point a bit later on. And so we segued into a topic, again, a virological topic, parasite-derived resistance. So what we were doing here was exploring a concept that had been developed with Q-betaphage in E. coli, which was by Sanford and Johnson. And the basic idea was that if you took a component of a parasite, expressed it in the host organism of that parasite, then for various reasons you might expect that the host organism would acquire immunity against the parasite. So you might expect, for example, that parasitism requires an interaction between a component of the parasite, component of the host, leading to disease. So if you prime the host by expressing in it a modified component of the parasite, that would block the receptor sites in the host. And so when the parasite is introduced to the organism, it's got basically nowhere to go, and you get resistance. Very simple concept. And in the early 1980s, again, my colleague Michael Bevan, working with me in the Plant Breeding Institute, had developed the technology for making genetically modified plants. And so the concept was very simple. We would take genes out of a virus, express them in the host plants of that virus in the expectation or the hope that we would get resistance against the virus. And the initial experiments we did were with potato-virus X. This is a virus that, an RNA virus infecting a whole range of different solanaceous plants, not only potatoes, and we transferred various genes, including the replicase protein gene, using Agrobacterium as the shuttle vector into the genome of plant cells. And then we tested the plants to see whether or not they were resistant against potato virus X. And this is where the luck comes into our story, in that we had a very unexpected result. When you make GM plants, when you make transgenic plants, you often get a range of phenotypes in the different lines that you've produced. And consistent with many of the other GM projects that we were doing at the time, we had some lines with the expected phenotype, or the, let's say, hoped for phenotype, the virus resistance, and some lines without the phenotype. So this was quite normal. But what was unexpected in this particular experiment was that the lines that were resistant against the potato virus X were apparently not expressing the transgene that we had put into the plants. We had similar lines that were expressing the transgene at a very high level, and these lines were fully susceptible to the virus. So this was odd, because normally you expect that some expression would be good, more expression would be even better. But here, apparently, we had no expression is good, a lot of expression is not giving us the result that we expected. At about the same time, there were a number of other people doing GM plant experiments, including some groups in Holland and the United States who were investigating flower pigment production. They were trying to make petunia plants that were more darkly pigmented than the lines that they started off with. So they were taking a chalcone synthase gene, adding it into the petunia genome, and in the expectation that they would have more pigment. Chalcone synthase is an enzyme in the pigmentation pathway. But what they got was this. So the transgene, the added transgene was silent, the endogenous chalcone synthase gene was silent. Sometimes they got this. So this is a pattern they referred to as the Cossack dancer pattern for reasons that you can maybe appreciate. We surmised that what we were looking at with our virological experiments was essentially the same mechanism as was being seen in these experiments with petunias. And we interpreted this as a phenomenon which we called RNA silencing. So we anticipated that the transgene DNA, the chalcone synthase DNA in the petunia plants, would be somehow priming the RNA silencing mechanism such that it silenced both the transgene RNA and the endogenous RNA. And in our virological experiments, we could use the same conceptual shape of the model. So the viral transgene DNA would be priming RNA silencing so that the viral transgene RNA would be silenced and the viral RNA would be silenced. The fact that we were using an RNA virus in these experiments was highly informative because it meant that the mechanism had to be operating at the RNA level. The petunia people were getting sidetracked into all sorts of DNA methylation stuff at the time, and so they were not able to see the way forwards as clearly as we could with our virological experiments. We then asked the question, well, how could you explain the specificity of this whole process? And we predicted that there would be an antisense RNA involved. So the transgene RNA perhaps would be producing antisense RNA, and this would be the specificity determinant of the RNA silencing mechanism. And indeed, that turned out to be the case. And Andrew Hamilton, a postdoc in the lab at the time and now a lecturer in Glasgow, did these hugely important experiments when he identified small antisense RNAs that were the specificity determinant of the RNA silencing mechanism. The first data he got like this, a little bit messy using RNase protection, but eventually he managed to get these very nice, tidy data using northern blotting. And so the RNA, the small RNAs that he was seeing, 21, 24 nucleotides, were the specificity determinant of the RNA silencing process. Now, there's an irony to this whole story because we set out to make an artificial virus resistance mechanism, but it turned out that what we were looking at was something that plants do anyway. They protect themselves against virus attack by using the same mechanism that was being activated in our transgenic plants. So it is a mechanism that is now quite well known. So double-stranded RNA is processed by DICER into small interfering RNAs, the RNAs that Andrew first revealed in the lab. And then the Argonaut protein is a second nuclease that uses these small interfering RNAs as a guide species so that it can target complementary messenger RNAs. So this is the evidence that this is a antiviral protection mechanism because these are two plants, both infected with an RNA virus, potato virus X. One of them is a normal plant. The other one is a mutant for one of the proteins in the RNA silencing pathway, and it's hypersusceptible to virus infection. And actually, there's an irony here. So my virological mentor at the time, Brian Harrison from Dundee, had introduced me to these experiments which were published in 1920s, showing an immunity phenomenon in plants. So the plants, when inoculated with a virus, initially develop symptoms, but then they recover. The recovered tissue does not contain virus, apparently, but cannot be superinfected with the virus that was used to prime the whole process in the first place. They can be infected with completely different, unrelated viruses. So there was a bit of a mystery here. You've got a highly specific virus resistance mechanism that is evidently operating in the plants, but of course plants don't make circulating antibodies and other components that give specificity to the immune system of animals. And it took 70 or 80 years before we could identify what the specificity determinant was in this whole process. Now, I'm not a person of regrets, but just looking back, what we noticed was that we could actually explain the non-coding RNA phenomena that we'd been investigating in the 1980s, and had we had a little more persistence. So what it turns out is that this non-coding RNA is giving rise to small interfering RNAs, and these, in this instance, are targeting a host enzyme for chlorophyll biosynthesis. So the plants, the infected plants, don't make chlorophyll because the viral RNA is silencing a host gene. Yes, if we'd followed it up a little earlier, been a bit more persistent, we could have been 10 years ahead of ourselves, but hey. Now, this is all very simple, and what you'd like to be able to do is to exploit this understanding of RNA silencing, and I hope that indeed we will be able to do this. So this is one of our current projects in the lab where we have an interest in a very serious disease in East Africa at the present, which is called maize lethal necrosis. It's a virus disease, RNA virus disease. It's actually two viruses infecting the maize plants. And it's spreading through Eastern and Central Africa, causing serious problems for maize growers there. And maize is central to the society in that part of the world. My student, Luke Braidwood, who's doing this project, visited East Africa, and he came back with the quote that in Kenya, maize is food, and everything else is just a snack. So this is a pretty important problem. And there are various approaches that you might try to address maize lethal necrosis, including resistance breeding, normal resistance breeding, but that's going to take a decade before you have the genotypes that are ready. And so what we're attempting to do together with collaborators in Kenya is to follow a biotechnology approach. And the idea here is that, first of all, you sample in the field, have a look at the diversity of the viruses that cause maize lethal necrosis. You then design artificial genes that will target conserved regions of those viruses so that you'll get broad spectrum resistance, transform maize plants with that, and then ultimately test plants for resistance. And this is a project that is ongoing at the moment, and I hope that within a couple of years we'll have resistant germplasm to test in East Africa. So this then is the simple mechanism. We've come to this understanding to some extent through serendipity. I think it's a very nice illustration of the elegance of nature in coming up with a defense system. And it's elegant because it provides really two features that are essential requirements of defense systems. So first, it's something that has got specificity. So a defense system with specificity is less likely to cause damage to the host, and so specificity is a requirement. It's also got an amplification component to it. So you start off with one double-stranded RNA, you end up with many small interfering RNAs, each of which is incorporated into a catalyst, the Argonaut protein. And so you've got amplification. One molecule can prime the destruction of many other viral RNAs in the system. So it's very nice. What I want to move on to now are the complications associated with counter-defense and also with counter-defense. And if it's not clear what I mean by that, I hope it will become clear after the next several slides. Now, in order to describe this next little story to you, I need to introduce you to one feature of RNA silencing, which is that it is not only a virus defense mechanism, it is more than that. We know that because we find evidence for RNA silencing in essentially all eukaryotic lineages. So we can infer that this is a process that existed in the last common ancestor of eukaryotic cells. And in the subsequent hundreds of millions of years since the evolution of this, the various eukaryotic lineages, there has been diversification of the RNA silencing pathways, and we can infer that because there is diversification of the proteins associated with RNA silencing. This is a phylogeny looking at the Argonaut proteins, the effector proteins of RNA silencing, and in each different lineages you see substantial diversity. This is the plant lineage here, this is C. elegans, humans and mammals are over here. So substantial functional diversification in RNA silencing. And one of the elements of diversification is the microRNA pathway. So these are endogenous RNAs which use dices and argonauts and they're small RNAs and they target endogenous genes. So they're part of the machinery that cells have, many eukaryotic cells, to regulate gene expression at the post-transcriptional level during normal growth and development. So this is one element that I need to introduce you to in order to make clear the story I'm going to tell you. The second element is that viruses encode proteins which are suppressors of RNA silencing. So this is part of the normal dynamic of defense and counter-defense that we're used to when we think about host-pathogen interactions. So a host develops a defense against the pathogen acquires a counter-defense measure. And the evidence for this in RNA silencing and plant viruses is that every virus that we've looked at has one or more genes encoding proteins which are able to somehow suppress the RNA silencing mechanism. This is one example here, not work from my lab. We first described this suppressor of silencing, but it was subsequently crystallized by Tracy Tanaka Hall, and she described how this protein forms a clamp around the end of the small interfering RNA and prevents it being recruited by the Argonaut protein complex. There are many other suppressors of silencing encoded by different viruses. They've all got rather boring names like P19 or 2A or P38. The one that I want to talk to you about today indirectly is P38. So this is a protein encoded by a class of plant viruses. And what this does is to bind to the slicer enzyme that is associated with microRNAs. So then two elements to this whole story. There are microRNAs in plants and there are suppressors of silencing. So the simple experiment that we did a few years ago was a mutation analysis in Arabidopsis. We wanted to say, well, yes, we know about the small RNAs and everything, but which of the 10 Argonauts in Arabidopsis is the one involved in antiviral defense? The experiment was very simple. You just look at mutants in each of the 10 different Argonaut protein genes and ask which of those mutants acquires hypersusceptibility to virus infection. So Argonaut 2 was the one example that we looked at with this particular virus, with turnip crinkle virus, where the mutant was hypersusceptible to the turnip crinkle virus. So that was nice. Argonaut 2 is the antiviral slicer for this particular RNA silencing branch. Another related observation was that in the virus-infected plants, Argonaut 2 is apparently induced. Now, this was a bit of a mystery. We didn't quite understand how it could be induced. And when you see something induced, you can either think in terms of a gene being switched on, activated, or you can think of repression of that gene being lost. And actually, we converged on the second of those ideas because it turns out that Argonaut 2 is the messenger RNA for Argonaut 2 is the target of a microRNA 403. So what you can anticipate is happening then in the cell is that turnip crinkle virus comes into the cell. It produces small RNAs, and these begin binding to Argonaut 2. The P38 suppressor of silencing is produced as the virus begins to accumulate. What this P38 suppressor of silencing then does is to prevent Argonaut 1 binding to microRNA 403. So this then releases the suppression of Argonaut 2. And so you get an increase in the messenger RNA for the antiviral slicer, that is Argonaut 2. And a test of this was to look in Argonaut 1 mutants, and indeed in Argonaut 1 mutants, the Argonaut 2 protein is constitutively produced. It doesn't need to be induced. So the interpretation of this then is in terms of what we refer to as this sort of zigzag scheme. So you start off with viral RNA, and this feeds into the simple RNA silencing pathway, so the amplitude of defense increases. Viral suppressors of silencing are then produced, and these block the microRNA-mediated suppression of Argonaut 2, so then you get more Argonaut 2, so then the amplitude of defense is increased. So this then is a little, let's say, complexity or element of complexity that we can superimpose on the simple mechanism that I described to you in the beginning. And I think the general message about this is, so often in host defense jargon, we talk about an arms race between the hosts and the pathogens. And I think this points out that the arms race concept is an oversimplification that really doesn't work in this whole system. And the way that I prefer to think about it is as, rather than an arms race, a coalition. And of course, within a coalition, you can have a number of different types of relationships. So you can have a cooperative relationship, let's say where if you anthropomorphize things where the virus and the host are sort of cooperating. And this it could result in a mild asymptomatic disease. And if you extend it to other microorganisms, it could end up, for example, as a mutualistic symbiosis. You could have a coalition which is a wary coexistence, let's say where there's a new combination of virus and host coming together. They haven't quite learned yet how to cooperate properly, and so there's some suppression of the virus by the host, but you get disease. And then you have other coalitions where one partner is doing its best to exploit the other one. So you either end up there with complete immunity, but that's not a very good long-term strategy because you then place a very strong selection pressure on the virus to overcome the resistance, or you end up with a sort of lethal disease. Now, you may want to sort of draw some parallels with political situations in these types of coalition here. And I'm a scientist, and so I'm not going to comment on that sort of thing, but I just want to make the point that this sort of defense, counter-defense idea that allows coalitions to happen between hosts and their pathogens, hosts and viruses, is a fairly general one. And in order to reinforce that point, I want to describe to you another set of projects that we've had going in the lab over the years. And in order to describe this project to you, I again need to introduce a couple of background pieces of information. So in plants, we have a number of different layers of defense, a number of different defense systems that operate against viruses. And one of these layers involves a protein-mediated innate immune system with a set of proteins that we refer to as nibblers. These are nucleotide binding site leucine-rich repeat proteins. Nibblers are a lot easier to say. What basically happens is that in a non-infected plant, the nibbler proteins are sort of folded up on themselves. And then once the pathogen, the virus, is introduced into the plants, there's a molecular recognition process in the virus that we work with, potato virus X, that is resisted by a protein called Rx. In the Rx potato virus X situation, the molecular recognition involves the coat protein. And following that molecular recognition, Rx unfolds. And then what it does is there's a change in the nucleotide binding status of this protein, and it activates defense. It's quite a nice system. Its limitation as a defense, or a feature of it as a defense system, is that it is quite specific. So in this instance, it's specific for strains of potato virus X that have a little TK motif at a particular position within their coat protein, but not against strains of the virus that have K and R, have lysine and arginine at those two amino acid residues. So this is a specific protein recognition mediated process. We've had, so there's recognition and response in this innate immune system. We've had a number of projects going on in the lab exploring, the specificity in this whole process, and we've been carrying out artificial evolution of different domains of the Rx protein in order to see whether or not we can get a broader spectrum resistance than is conferred by the genes that exist in the natural potato and potato relative populations. And we use an error-prone PCR strategy for this. This is not the subject of my talk, so I'll sort of gloss over it just to say that yes, we can. And as in many things in science, if you try to mimic nature, then you get a better outcome. So rather than trying to get what you want in one go, a big step in one go, we've found that a sort of Darwinian approach here is much better. So if we do several small steps, we can get a better, broader spectrum resistance than if we try and do it in one go. So this is Rx. This is protein-mediated defense. It involves nucleotide binding site leucine-rich repeat proteins. And the one point I want to make about this level of defense, which is that like many defense systems, there is a fitness cost to the plant for carrying the proteins associated with the system. And there's good evidence for that, not specifically with Rx, but with other proteins in this class. And this idea that this immune system has got a fitness cost features in a moment. The second element that I'd like to introduce you to associated with this aspect of our story is another of these diversifications of the RNA silencing pathways. So this one involves microRNAs, or it could involve microRNAs. So it's a system, an amplification system involving secondary small RNAs. So what happens in this system is that you've got a primary small RNA. It can be a microRNA, or it can be a small interfering RNA bound to an Argonaut protein. It finds a target RNA. And the target RNA, rather than being simply degraded, becomes a substrate for an RNA-dependent RNA polymerase. So that means that then it is converted into a double-stranded RNA. A double-stranded RNA can then be diced into secondary small RNAs, and those secondary small RNAs can then go and target other messenger RNAs within the cell. So it's a system with an amplification potential to it. You start off with one small RNA, you end up with many small RNAs, and each of those can go away and target other genes. So you can think of regulatory cascades in connection with this secondary small RNA pathway. Here's another element of luck. So one of the things that we were interested to do was we started, for reasons I can't go into, started working with tomatoes rather than Arabidopsis and tobaccos and various other things. And we wanted to ask, in tomato, which messenger RNAs are associated with these secondary small RNAs. And it turns out that we could see very good evidence that the nibbler protein messenger RNAs are involved in secondary small RNA production. And we could also identify the microRNA that initiates the secondary small RNAs. It happens to be called microRNA 482. So this is then the two areas of interest in the lab converging here. What we found was that microRNA 482 exists as a small family of microRNAs. And the variation between the different family members corresponds to wobble position of the triplet codons in terms of the targeting specificity. It also turns out that microRNA 482 targets the P-loop motif, so a very highly conserved motif within all of the messenger RNAs for these nibbler proteins. So what this means is that with the microRNAs targeting the same protein coding motif, but having variation at the wobble position, is that a small family of these microRNAs can target a very high proportion of the nibbler protein messenger RNAs. And we conservatively estimate that it's something like 30%. I suspect that it's a lot more than that. So then we've got essentially a system in which the protein-mediated innate immune system is downregulated by a microRNA in a non-infected plant. So what happens then when a plant gets infected? Well, it's essentially the same as with the Argonaut 2 story. So the viral suppressors of silencing are being produced. These prevent microRNA 482 from downregulating the innate immune system, so then you get upregulation of the nibbler protein messenger RNAs and more abundant nibbler proteins. And we've got evidence that this indeed happens with virus infection, and there's emerging data now, certainly with oomycete pathogens and bacterial pathogens of plants, and I suspect there's some straws in the wind with fungal pathogens as well, that these pathogens too produce proteins, effector proteins, that are suppressors of RNA silencing. So this is then a system which could explain how it is that when a plant gets infected with a pathogen, that it can upregulate its protein-mediated innate immune system. So why would a plant want to do this? Well, it's all to do with the fitness cost of carrying these genes. And the evidence for this is when we look at the number of nibbler protein genes within a genome, and whether or not the plant genome encodes or does not encode microRNA 482. What we find is that all of the genomes that we've looked at with very large numbers of the nibbler protein genes, these are the ones that produce the microRNA 482. If the plants have small numbers, few nibbler protein genes, then they can afford to get rid of the microRNA 482. And so what we're thinking is that this is a system that allows a genome allowed to, let's say, accommodate the fitness cost associated with diversity in this innate immune system. And so here we've got RNA silencing taking us in a slightly different direction from where we started out. So rather than being RNA-mediated immunity, this is protein-mediated immunity. We've got microRNAs as fine-tuners of the innate immune system, and then we've got pathogen-derived suppressors as fine-tuners of the fine-tuning system. And the sort of exquisite tuning that you need if you're going to have a coalition rather than an arms race. Now, I just want to take you through one final part of our little story here. And in order to lead you into this, I'll describe a technology that we developed very early on when we were getting interested in this RNA silencing business. So what we understood was that when a virus goes into a cell, you get small RNAs being produced corresponding to the viral genome. So then what we thought was that if we added into a viral genome a small fragment of a host gene, that small RNAs would be would be produced that correspond to that insert. And so we would get a symptom on the infected plants, which phenocopies a loss of function mutation in whichever gene is represented by the insert. And the little panel on the bottom right-hand corner there shows you indeed that that does happen. So this is the flower from a Nicotiana species on the left infected with a control virus, and on the right infected with a virus carrying a fragment of a gene called leafy. And leafy is a gene that is required for flower development. In a mutant plant, you get leaves rather than flowers, and that's indeed what we've got on our virus-infected plant here. So, virus-induced gene silencing, a very nice technology. It's proved to be useful to us and others as a tool in functional genomics. Now, over the years, we like sort of fiddling around a bit, and one of the questions that we wanted to do was to say, well, what happens when you fiddle around with this VIGS, virus-induced gene silencing system? So I've just shown you how if you take part of the coding sequence for a gene and put it into a virus, you'll get post-transcriptional silencing and you get suppression of the target gene. We wanted to say, well, what happens if you put part of the promoter sequence in? And so we did this on plants that were carrying a GFP transgene. So this is a traffic light gene. When the gene is on, the plants are green under UV light. When it's off, the chlorophyll fluoresces red under UV light. So here's the control. It's on in the plant infected with a control virus. It's off in the plant infected with the coding sequence virus. Interestingly, it was also off if we added in part of the promoter sequence into the virus. And when we looked in these plants in a little more detail, what we could see was it was not turnover of the RNA which was being accelerated, but transcription of the gene which was being suppressed in the plants infected with this promoter sequence construct. But this was not the only element of interest in these experiments, because when we looked at the progeny plants from these experiments, what we saw was that with the promoter sequence construct, the silencing effect persisted into the next generation. The virus is not transmitted into the next generation. This is not a seed-transmitted virus. There was no virus in the progeny plants. With the coding sequence constructs, the silencing effect was lost in the seedlings, shown here, of the next generation. But with the progeny of the promoter sequence constructs, the silencing effect persisted. And it persisted in the progeny of the progeny, and the progeny of the progeny of the progeny, and so on and so forth. So what we've got here is a very clear evidence for a transgenerational epigenetic effect. Virus-induced gene silencing, VIGS, was associated with methylation of the target DNA, we and others have done a bit of molecular biology stamp collecting, and we've now got quite a good understanding of the mechanisms that underpin this RNA-mediated epigenetic silencing. Just parenthetically, this also suggests to us a strategy for making an epigenetically modified plant. So what you might do here is to start with a plant in which a gene is on, that, let's say, in order to improve that plant as a crop, you would like to turn that gene off. So what you then do is to design a virus carrying RNA that will target the gene you wish to turn off. You'll infect the plant with this. The virus spreads through the plant, does its gene silencing stuff so that eventually in the infected plant, the gene is off throughout the plant, and the plant will produce seed, and you can sow out the seed and ask what happens to those seed in the next generation. We've got examples with several different target genes now, both from our work and in the literature, that the silencing effect persists into the next generation. So it's stably inherited. Now, it's not only, maybe I've got a slightly mischievous streak and frustrated by the difficulty of getting genetically modified plants into the field. I would like to ask the question, well, can we put epigenetically modified plants into the field? So according to the current EU legislation, although I suspect that it may change, epigenetically modified plants produced in this manner would be exempt from the GM regulations. And so we'll be setting up some field trials this summer in order to explore the system. So we'll apply to have epigenetically modified plants going through the whole process. I suspect that we will be converging in on this whole epigenetic topic in other aspects of our interest in disease and defense against disease, because there's good evidence in plant systems, certainly, that defense can be primed. So if you infect a plant, this is not our work, this is Yuri Anton's group work. If you infect plants with a Pseudomonas species, the defense systems in those plants is primed, and the defense priming persists from one generation to the next. And there's maybe reason for thinking that the RNA-directed epigenetic mechanisms that I've described to you are somehow implicated in this priming system of defense mechanisms. So I think that this will be another element that we will eventually converge upon as part of our interest in RNA silencing. So that, I think, is where I would like to close. These set of headings, I think, are my summary slide, if you like. So we came to work on RNA silencing by accident, but we've enjoyed the 20-odd years that we've been involved in this topic. It's a very simple mechanism in principle, but the diversification of the RNA silencing mechanism opens up understanding of the complex ways in which plants and viruses, plants and microbes can interact with each other, and how these interactions can have very different outcomes. And it's also taken us into this whole topic of epigenetics. So with that, I will close. The work I've described to you is work that's involved a great number of people in the lab over, as I say, 2 decades, really. But these are some of our current lab members and collaborators. So I'd like to thank you for your attention, and also to thank the Society for the very great honor of allowing me to deliver this, or giving me the privilege of delivering this lecture. I described how I came into plant virology as a complete novice. I'd got no training or background in virology, in microbiology, and the Society was very supportive. It was a great place to come and meet other virologists and give talks as we entered this field. And so I've been grateful to the Society for a long time. It's a real privilege to have this award from the Society. Thank you very much.